For the next 3 lectures, we'll talk about specific techniques that are common in labs, and today in particular, we'll discuss PCR. So a quick show of hands. I don't have a slide for this, but who has heard of PCR before? Pretty much about everyone. And one of the reasons for that, I asked the same question 4 or 5 years ago, and I had only 10%, 20% of the hands go up. Then COVID happened, and everybody knew what a PCR was, right? So we all know what a PCR is. Now, if I asked any of you to describe it for me in a few words, what would you say it is? Awesome.
Kind of like breaking down DNA and then trying to replicate it.
Breaking down DNA and trying to replicate it, okay. Gene amplification, okay. Any other ideas?
Did you say
Gene cloning, okay. It's so fun because you guys are making the list that we will discuss Tuesday. All of you mentioned one way or another uses of PCR, right? And we will see how we use PCR for all of those things, for cloning, for amplifying genes, and then breaking down things and making many copies or replicating those things that we broke down. So super generically speaking, PCR is simply making many copies of a sequence of DNA. That's all it is. What we do with that later can take on a number of shapes and flavors, and we'll discuss several of them, but next week. So before PCR, if biologists wanted to make many copies of a given DNA sequence, they would either extract that DNA sequence from a big organism, and these were very crude approaches, but that's what people did, right? So it's funny, this was even before my time, just to make it clear, but you would walk into a molecular biology lab, and one of the most common pieces of equipment was a household blender. That's what people used to just blend up organic material. And then you would find in this ocean of DNA the sequence that you were interested in. A little later on, when I say we, I wasn't part of any of that, but the collective we, when we figured out how to introduce DNA into bacteria, we would use those bacteria as mini living factories to replicate themselves, and in the process replicate the DNA that we introduced into them and make many copies of those DNAs for us. So the approach became something like you would introduce your DNA of interest, which is depicted as that red line, into a bacterium, let the bacterium do what they do best, which is replicate many times, and then extract those DNA sequences from them. So those were the approaches before PCR. Early to mid 80s, someone comes along and thinks, well, can't we replicate the bacterial replication, no pun intended, in a test tube? Can we just do this DNA replication in a test tube instead of having to depend or rely on these living organisms? And essentially, what these individuals did was essentially learn from what we know about DNA replication in living systems in cells and see how that could be replicated in a test tube without a living organism around it or around that DNA. So in Canvas, there are a series of links to chapters in your various books that deal with DNA replication. If you need a refresher on all of that, I would recommend that you take a look at those. We'll go through the major steps in DNA replication briefly here, but more importantly, what we'll do is we'll compare what cells do inside of them to replicate DNA with what we do in tubes for PCR, okay? So the very first thing that a cell needs to do if it wants to replicate DNA is to separate the strands. Right, we're talking double-stranded, double helical DNA. It first needs to separate those strands, and it does that with the help of enzymes that we call helicases that will, using energy, separate the strands of DNA, or as we say in jargon, unwind that DNA. How do we do that in a test tube? Anyone knows? How can you separate strands of DNA in a test tube?
Like 95 degrees Celsius, so
Heat, that's the key words. So that's exactly right. The bonds that are keeping the two strands of DNA together are not covalent, right? And even though the DNA is a very stable double-stranded helix, those bonds are collectively strong, but individually kind of weak. So if you increase the temperature of the system, you will force these strands to separate. And that's exactly what we do in PCR. We use heat. And a key difference between what cells do with helicases and what we do in PCR with heat is that if you apply enough temperature to the system, you can separate the entirety of a DNA molecule into its two strands, as opposed to helicase, which will work locally. Helicase will open up very restricted regions of that long DNA molecule. Whereas heat, if we bring it up to about almost boiling, 95 degrees Celsius, you can separate both strands in their entirety. So far, so good? Cool. So after cells have separated the strands of DNA, it turns out that unlike RNA polymerases, those enzymes that make RNA molecules based on a template DNA strand, DNA polymerases can start from scratch. They cannot. They need to start from a 3' hydroxyl group that is already there for them, right? They need to have, I know it sounds a little counterintuitive, they need to have a bunch of nucleotides already present for them to initiate the synthesis of new DNA. Sounds a little weird, but that's how evolution did it, right? So they cannot start from scratch. They need a pre-existing sequence. RNA polymerases don't. So there is this very special family of RNA polymerases called primases that will go into these regions that were unwound by helicases, and they will start from scratch the synthesis of a short RNA sequence based, as always, on the rules of base complementarity and anti-polarity that these polymerases normally use to make a short RNA sequence that we call a primer. That's why the enzyme is called a primase. Okay? So that's how cells do it. They will use an enzyme called a primase to make a short RNA primer. How do we do it for PCR? What do we do? We introduce our own primers. We have machines that, if you will, took the place of primases that will make primers for us, and they will stitch together a series of nucleotides that will be complementary to a specific region of DNA that we're interested in amplifying. We'll go through that in a second. But essentially, we don't use enzymes to make primers for us. We use machines that will make primers for us.
Is that what CRISPR does?
No. Yeah, no. With CRISPR, we will talk about CRISPR in a few weeks, but no. Short answer is no. So in PCR, we use human-made primers, and those will, biochemically speaking, serve the same role that RNA primers serve in DNA replication within cells. They will go find their complementary sequence in a template single-stranded DNA molecule and bind to it. And it will offer that critical 3' hydroxyl group that any DNA polymerase would need to pick up the synthesis of that DNA molecule from there on. Okay? I'll introduce here a little bit of jargon. Let's say that the sequence of DNA that we're interested in amplifying is located in this region here. The top strand in this cartoon is 5' to 3'. The bottom strand is 3' to 5'. Synthesis of DNA, like synthesis of RNA, proceeds in the 5' to 3' direction, which means that in the bottom strand, the one that is 3', 5', the template will be read from left to right. The new DNA will be synthesized 5 to 3. The top strand will be read, quote unquote, backwards from right to left, right? So the synthesis will still proceed 5 to 3. Does that make sense? So because of this idea that one primer, the synthesis will go left to right, and using sort of this mindset of writing left to right, as you would find in Romantic languages and Western languages, we call those the forward primers. And the primers that will, again, I can't emphasize this enough. It's not like DNA synthesis is going backwards from those primers. It is still going in a 5' to 3' direction, because that's the only way that DNA polymerases can function. But from our perspective, they're going backwards, and we call those reverse primers. Okay? So from here on out, you might hear me say, we're not going to go into the details, but you might hear me talk about forward primers and reverse primers, forward primers and reverse primers. What I mean by that is anytime that you want to amplify a specific region of DNA, you need primers that are flanking that region. One of them looking in from the left, I guess, we call the forward primer. The one looking in from the right, we call the reverse one. Yeah? And it is always important to have both. We'll see in a second why. All right, so in cells, primases already made those primers, and now it's the job of DNA polymerases, and in particular DNA polymerase 3 in eukaryotic cells. Prokaryotes have only one kind of actually, no, they still have several kinds. So DNA polymerase 3 is the one that, once that RNA primer is present in the unwound DNA, will pick it up from the end of that primer and continue adding nucleotides using the rules of complementarity and anti-parallel orientation to synthesize the complementary strand of DNA to each of the templates. We're not going to go into a lot of the details, but if you review those chapters on DNA replication, you'll see that there is this issue with the way that what we call the replication fork moves on DNA, right? And how one of the strands can be used, the one that we call the leading strand, can be used as a template continuously. So a primer is made, and from that point onwards, the DNA polymerase can add one nucleotide at a time without any interruptions. On the other strand, there's this phenomenon that has to do with the orientation of the strand, that as the DNA is being exposed, the DNA polymerase is forced to move backwards. And because it is forced to move backwards, up in the opposite direction to the way that DNA is being opened up, when new DNA opens up and offers new DNA template sequence for DNA polymerase to work with, this DNA polymerase went backwards. It went in the wrong direction, so it has to detach, reattach, and start again. So while on one strand, synthesis of new DNA is continuous and smooth and uninterrupted, in the other strand, synthesis of DNA is fragmented. Right? If you hadn't seen this before, you hadn't ever heard about this before, hit those chapters and everything will be explained there. So bottom line is, in cells open up the DNA, make the primers, and then let DNA polymerase 3 take it up from there, and DNA polymerase will start making DNA either continuously or interruptedly, depending on what strand they're in. How about PCR? In PCR, it's not much different. The only difference is that we don't use just any DNA polymerase 3. We use a very special family of DNA polymerases that were extracted from very peculiar organisms that live in very, very hot environments. Right? Generically, you will see these special DNA polymerases referred to as Taq polymerases. That's in honor of the very first of these enzymes that was used in first PCRs. And Taq is really short for Thermophilus aquaticus. So this was a bacterium that lived in very hot springs. And the reason why we use Taq polymerases is because, as we will see shortly, we will subject PCR reactions to various rounds of heating up. And any of the polymerases in our bodies right now, if you heat them up and then you let the mixture or the reaction cool down, our polymerases would get destroyed from that heating up and cooling. The whole heating up of our polymerases would make them lose their shape in space and therefore lose their function. These organisms evolved in very hot environments, so for them, heat is the most natural thing. You have them in a tube and you heat that tube to close to boiling temperatures. For them, it's like, I'm back home, right? And they don't lose function with these repeated rounds of heating and cooling. Yeah?
Is there a certain heat that they'll break down? Or
Yeah, when you go over boiling, when you go grossly over boiling,
so
yeah.
they're just heat tolerant?
Yes, yeah. They're not like heat completely is resistant, impervious. Yeah, no, they're heat tolerant. In fact, their temperature range is much higher than us. Like us, or like our DNA polymerases, these enzymes also have a temperature range. It's just shifted to hotter temperatures. Yeah? So the other key difference between PCR and DNA replication in cells is, and now I would like to go back to what I emphasized at the beginning, since the original heating up of the sample separated both strands completely, now the Taq polymerase that we add to our PCR reactions, our PCR mixtures, doesn't care anymore about the direction of opening of DNA. DNA is fully open. So all of the DNA present in the sample is exposed as a template single-stranded DNA molecule. So they don't have to worry anymore about, oh my God, I'm going in one direction, but DNA is opening up in the opposite direction. I have to let go here, go catch up. They don't have to worry about that. They can start from where they started, and then everything in front of them will be open and available. Did that make sense? So for PCR, there is no such thing as continuous and interrupted polymerization. It's all continuous on both strands. Okay? That comes handy in the next couple slides. So in a cell, DNA polymerase 3 did its job, and after DNA polymerase 3 did its job, you have that what used to be one double-stranded DNA molecule now became two double-stranded DNA molecules. In each of them, one of the strands was from the original DNA molecule, if you guys recall your semi-conservative DNA replication. Right? So for each of the new molecules, one of the strands is old. The new DNA strands are a mixture of these long new DNA strands that were made uninterruptedly by DNA polymerase on the leading strand, and then these fragments that result from DNA polymerase having to let go, go back, and make some more, and then let go, go back, make some more. Right? So it's just a consequence of how DNA replication works in cells. So that's one, that the new DNA strands in DNA replication are fragmented by nature. The other feature about new DNA strands in DNA replication is that they all have a little bit of RNA in them. Primases make RNA primers in cells. We, with our machines, we can make human-made DNA primers, but cells make RNA primers. So now, new DNA molecules are not really totally DNA. They have little bits of RNA in them. So the way cells solve this is by introducing DNA polymerase 1 and an enzyme called ligase that work in tandem. DNA polymerase 1 will go and fit into any of these gaps that exist between a fragment of DNA and the primer sequence of the next fragment. They will fit themselves in there, and then they will do something very peculiar. They will start adding DNA nucleotides to the back and chewing RNA nucleotides in their front. So they move one position over by chewing off one of the RNA nucleotides and adding the corresponding DNA nucleotide behind them. They chew off an RNA nucleotide, they add a DNA nucleotide behind them. More technical terms, they chew off a ribonucleotide, they add a deoxyribonucleotide. Chew off the ribonucleotide, add the corresponding deoxyribonucleotide. And in doing so, they replace those stretches of RNA sequence with DNA sequence. So that solved the first problem. All of the bits of RNA that were embedded in these new DNA strands have been exchanged for DNA. So we're good. The thing that DNA polymerase 1 cannot do is close that gap. At any time when it chews off a nucleotide and it adds one on its back, it cannot link the two together. So after DNA polymerase 1 did its job, there is still some gaps in between these fragments. Now they're all DNA because the RNA has been exchanged. They're all DNA, but there is still some gaps in between nucleotides. And that's where ligase comes in and just like the name indicates, ligates both ends of neighbor nucleotides together, making that strand continuous. Yeah? How do we replace DNA polymerase and ligase in PCR? I see one head nodding. We don't. That's right. So, that's just a picture to show what I said before. The DNA polymerase will take out all of the RNA primers that were denoted by green lines in that cartoon, and then ligase comes in and ligates the ends. In PCR, there's no need for DNA polymerase 1 and ligase because DNA synthesis is never interrupted, so no gaps between fragments, and there's never been any RNA involved in DNA synthesis since the primers that we added made by human-made machines were made of deoxyribonucleotides, not ribonucleotides. Okay? So there's no need for DNA pol 1 and ligase. Yes? Let me go one back slide. So imagine that DNA polymerase 1 sort of fit itself in between a previous DNA fragment and the next. Every new DNA fragment starts with a little bit of RNA because that's what primases make, right? So what DNA polymerase 1 does is it replaces all of those ribonucleotides for deoxyribonucleotides. It will go through the primer and then some more, and then at some point it will fall off, right? I personally not sure exactly how this process goes, but it's not like it rewrites everything. It rewrites over the primer and a little more, and then it falls off. When it falls off, it didn't really finish the job of linking chemically through a covalent phosphodiester bond the last nucleotide with the next. So there's a little bit of a gap left, like there. So ligase comes in and just joins the two together. Yeah? And it does the same here. And now you have two new strands of DNA that are whole and pure in the sense that they're all made by deoxyribonucleotides. There are no ribonucleotides embedded anywhere. In summary, in the old days, if we wanted to get many copies of a given DNA sequence of interest, we would rely on living organisms. And let's say that the sequence that we're interested in is boxed in this yellow box here. We would rely on those organisms to replicate the whole of the DNA, including our sequence of interest. And then it would be our job to extract that sequence from the rest. Right? Nowadays, with PCR, we simply restrict DNA replication to a very specific region that is determined by the identity of those forward and reverse primers that we use. So if we want to amplify the region indicated by the same yellow box here, all we need to do is carefully design primers, a forward primer and a reverse primer that are at the boundaries of those sequences that we're interested in amplifying. And then we proceed with PCR, and the only DNA that will be made over and over and over and over again is the one that corresponds to that region only. Which obviously facilitates the isolation of these DNA molecules later on, because we don't have to isolate anything. All of the copies that we got, those many copies of DNA, are the ones that we're interested in. So there is no need to isolate them from anything else. All right, now let's go through, and I'm going to do this. That should not have been there. Let's see. I'm going to go through the steps of PCR in a bit more detail, like I've done for some previous mechanisms we covered in class. I would invite everybody, particularly if you're a visual learner, to engage with videos that show PCR in a much more dynamic and movie kind of approach. There's hundreds of them online, and they tend to be pretty good. So, but what I wanted to do here with you is home in on some specific details that are often missing from those overview videos of PCR, right? And some of the details that matter. So let's first start with the parts. What is it that we require for a PCR? Any PCR will require the double-stranded DNA template that contains the sequence of interest, right? We want to amplify a sequence, but that sequence needs to pre-exist our PCR. We cannot do PCR and generate a sequence magically out of a hat. The sequence PCR uses pre-existing sequence and will make many copies of it. Does that make sense? So PCR will not create sequences, as we would say, de novo, meaning from scratch or from scratch. Of course, we will need a supply of the 4 DNA nucleotides, a heat-stable DNA polymerase, most commonly Taq polymerase from Thermophilus, that should be. So that's Thermophilus aquaticus. And then, perhaps most importantly, the 2 different single-stranded DNA primers flanking the SOI, the sequence of interest. So, what are those primers like? I keep talking about these primers. These primers have features. A, they have to be reverse complementary to the region of DNA that we want to target, right? So this forward primer has to be reverse complementary to this particular region in the template DNA, and the same for this other primer. They have to have the right orientation, of course. And then a question that I get often is, how long should these primers be? And practice has it that primers are typically somewhere between 15 and 25 to 30 nucleotides. The shorter a primer is, the less specific it will probably be, because the chances of finding a sequence that is complementary to that primer somewhere in the DNA of an organism is proportional to the number of nucleotides that you have in your primer. So if you had a primer with 1 nucleotide, what would be the chance of finding an A in DNA? Well, 1 in 4, right? What would be the chance of finding an AT? Well, 1 in 16, but in a DNA molecule that has 3 billion nucleotides in it, you will find many ATs. Now, if you have a sequence that is 20 nucleotides long, the chance that that primer will find its complementary sequence randomly in DNA decrease drastically, okay? So length of primers is typically somewhere between 15 and 25 nucleotides long. And then, of course, a buffer solution with the salts and buffering molecules, pH, and so forth. Yes?
You said the shorter the primer, what?
The shorter the primer, the higher the chance that that primer may go and bind to a place in DNA that you're not really interested in, right? So we save shorter primers for when we have smaller target DNA molecules. So, for instance, we may use shorter primers with prokaryotes because they have less DNA in them, so we can play the odds and get away with smaller primers, because even though the chance of finding a target sequence by chance, randomly, increase, that prokaryotic genome is smaller, so we may get away with it. Now, if on the other hand, you go and use short primers in a species that has a genome with 10 billion nucleotides in it, then your chances that this primer will go bind elsewhere, not where you want it, but somewhere else, increase. Do you know what PCR stands for? Polymerase chain reaction. Because as we will see in a second, it's like a chain reaction like those in nuclear blasts, right? Where the one step in the reaction will create the substrates for the next step in the reaction, which will create the substrates for the next step. So it's a chain reaction, and it explodes into many, many, many copies of DNA. I personally wish that we save that C for cyclic, because PCR is ultimately a polymerase cyclic reaction. We're subjecting this DNA amplification reaction to several cycles. We're doing several times the same thing. So whichever way you prefer to remember it, what we'll discuss now are PCR cycles. So in a typical PCR, your cycle would start with that heating that we discussed before. We will bring our sample that has the template DNA, the deoxyribonucleotides, the Taq polymerase, and the primers, all in the same mixture. We heat the whole thing up to about 95 degrees Celsius, a few degrees short of boiling, okay? And we do that for anywhere between a few seconds to a couple minutes. What that will do is what we call denature the DNA template. It will separate both strands of the DNA molecule that we're targeting. Remember, we're not trying to amplify the whole thing. We're trying to amplify a region within this long DNA molecule, but we need to denature the whole thing first. In this cartoon, the two blue lines indicate that long double-stranded DNA template, and the black and red arrows indicate the forward and reverse primers, all mixed together. Okay? So when we denature the DNA, what we'll see is that now both strands of DNA are basically bathed in a solution of primers. But nothing is binding to anything while this thing is at 95 degrees, because that is so hot that the hydrogen bonds that typically keep nucleotides binding to each other in a double-stranded DNA molecule or a primer binding a DNA strand, those hydrogen bonds cannot tolerate these high temperatures. So everything is swimming around by themselves. The second step in a typical PCR cycle is what we call annealing. And in annealing, what we do is we lower the temperature to somewhere between 55 and 60 degrees Celsius, and we do that for about 15 seconds. When we do that, what's going to happen is this. What do you think is going to happen to the original DNA template strands when we lower the temperature to about 55? Could they come back together, right? You would think that they do come back together. Except that since we kept them so hot for so long, they separated and they started kind of drifting apart. So the whole coming back together would depend on them finding themselves and realigning themselves, which would eventually happen because once you get a few nucleotides doing it, and if you have them properly aligned, it's like a zipper, right? So it happens really quickly. But that initial finding one another in the right orientation might take a while.
So could it find like others to
Bind to?
bind to?
Like what? Like the primers. Exactly. These strands are surrounded by primers. So when we cool down the mixture for a few seconds, what they will bind to are the primers. The forward will bind to the 3 to 5 prime strand. The reverse will bind the 5 to 3.
So even though, like, the primers, they would be in the solution the entire time, even while it's being cleaned up to denature it?
Yeah, because they're there all the time. When you denature, everything is separate from everything.
Bases won't lose their function or anything like that? Or
Nope.
start to-
No, because one thing about, and this is key, that's a good question. So one thing that is key to understand PCR is hydrogen bonds that keep strands together and primers and target DNA sequences together are thermolabile, right? So they're a little weaker, they're not covalent bonds, so they cannot stand all that heat, they separate. But phosphodiester bonds, on the other hand, much stronger. So those you're not gonna break with heat. So with heat and cooling and heating and cooling and heating and cooling, you separate strands from each other, but you're not going to separate nucleotides from one another. You're not going to break those phosphodiester bonds linking the nucleotides in a strand or a primer. Tiny primer, 18, 20 nucleotides in length, how is it going to find its target sequence in DNA? Well, any given primer may never find it. But remember that these things are surrounded by primers. So flip that image around and think of a template strand being surrounded by primers that will match that region, right? So it's just a matter of nanoseconds or microseconds until the primer binds to its target sequence. Then we proceed to another round of elevating the temperature a little bit. We won't go to 95, otherwise we will re-separate everything. We go to what is called an elongation or extension temperature, and that is typically 72 degrees Celsius. And that 72 degrees Celsius seems like very arbitrary, and it is very arbitrary, is the optimal temperature for Taq polymerase. Is the temperature that Taq polymerase likes the best. So we give it the best conditions for it to bind the free 3' end of the primer and initiate the synthesis of new DNA, of a new DNA strand from there on. I'll give everybody a few seconds to think about this interesting question. We say that we need to lower the temperature to 55 or 60 to let the primers anneal their target DNA, right? And then we say that we elevate the temperature to 72 for Taq polymerase to do its job. My question is, what will happen to the primers when we raise the temperature to 72? How come those primers don't fall off at 72? At 95, they will certainly separate, right? Before I was making the argument that you need 55 to 60 for these primers to bind DNA. And I'm telling you, it's okay, you can raise it to 72. Why didn't we lower it to 72 in the first place then?
Because we want to be sure.
Ah, okay, interesting. I mean, we want to make sure of what?
That they bind. Yeah, so like by lowering it past.
Okay. So one argument here is they could have bound to the template DNA at 72, but at 55 we're making sure that they're bound. Interesting argument. Any other ideas? It's a little more nuanced than that. So at 72, they wouldn't bind. They do need the temperature to be around 55 to 60 typically for a primer that is about 20 nucleotides in length to bind their target DNA sequence. At 72, they wouldn't. So how come they remain bound at 72? Because, and this is a slightly more elaborate image of what really goes on. When you do annealing, I told you that within nanoseconds or microseconds, the primer would bind to the target sequence. The rest of the time, those 14 seconds or so, Taq polymerase is at work already. Why? Taq polymerase works best at 72, but it works just fine at 55 or 60. And it will start adding nucleotides, and it has 15 seconds to add nucleotides. And there is a calculation that says that a properly working polymerase will add about 1,000 nucleotides a minute. So if we do the math, we're talking a little over 100 nucleotides in 15 seconds. So by the time that we raise the temperature to 72, it's no longer a 15 or 20 nucleotide primer bound to DNA. It's the primer plus anything else Taq polymerase made during those 14 seconds. Let's say, for argument's sake, 100 more nucleotides. So now we have a molecule that is 120 nucleotides long bound to DNA. And those you're not going to separate by bringing the temperature up to 72. You would need the 95 again. All right, so we let Taq polymerase do its job first at a lower temperature, one that it might be going a little slower. Then we raise the temperature to its optimal temperature. It goes really fast, and it will make a lot of DNA from the primer until we stop it. And we will stop it when we reheat this thing to 95 and everything kind of falls apart, right? So for a second, and just for understanding the process of PCR from a theoretical point of view, after one PCR cycle, we would end up with two hybrid molecules that are made of each of the original DNA strands, and I'm talking the whole thing, the whole chromosome, right? And another DNA molecule that goes from each of the corresponding primers till as far as DNA polymerase or the Taq polymerase could go during that minute of extension, right? So we let it go for about a minute. I told you that it can go up to 1,000 nucleotides or so during that minute. So now we'll have a DNA molecule that goes from the primer in about 1,000, 1,100, 1,200 nucleotides in the other direction. Now, if you recall, that is way beyond what we really wanted. Remember, we wanted something that is between those two primers. We wanted something that is way more restricted. And these things are going way over. But that is what we would have after one PCR cycle. And I like to call this, you're not going to find this in absolutely any books or anything. I like to call these beyond pecans. Don't age yourselves by immediately understanding the reference. Some of you should say, oh, I wasn't even born. Some heads shaking, I like that. Anyway, so I like to call these things, and this is totally in-house, beyond pecans, because they go beyond what we really want, which we will call in a few slides amplicons. What we really, really want are DNA sequences that are amplified many times, and we call amplicons. These things are going beyond that. So these are hybrid beyond pecans. Let's go for that second cycle of PCR. I just told you, all we do is we raise the temperature again to 95 for a few seconds to about a minute, or a couple minutes. Everything melts again. And when I say melt, I mean it, that's the jargon we use. All the DNA molecules present in that mixture will separate because no hydrogen bond, no matter how long those molecules are, can take that, right? So they separate. And now we have the original single strands of DNA, these beyond pecans, and everything is bathed in primers. So when we go for annealing, now the primers will very quickly find, or you can think of it the other way around, each of these original DNA strands and the beyond pecans really quickly find a target that will bind to them when we lower the temperature to about 50 to 60 degrees, right? So we see the forward primer binding to the beyond pecan that is going backwards, the reverse primer binding to the original strand that is going forward, and vice versa. Yeah? So the targets found their target sequence again. And once again, Taq polymerase will immediately bind to these primers and initiate synthesis, even though we're not at their optimal temperature. It's good enough for Taq polymerase to do some synthesis. It will do that, and by the time we raise it to 72 for about a minute, those new DNA molecules will not separate from their template because they're long enough now that they can withstand the 72, and we can let extension proceed for about a minute. So what I want you to notice, and this is something that those videos out there cover relatively well, but what I want you to notice real quick is the original strands, the very original strands of DNA present in the sample will always serve as templates for beyond pecans. Every single time that a primer binds to them, they will continue synthesis for as long as we let that Taq polymerase go, which is usually beyond what we need. Okay? So the original DNA strands will always serve as templates for new beyond pecans. But the beyond pecans that were produced in the first cycle of PCR and that are serving as templates in the second cycle are very peculiar in the sense that when the, here, I'll show you here. When the new DNA is being synthesized over a beyond pecan, there is no infinite template. The beyond pecan had a beginning where the first primer was, and when the DNA polymerase, the Taq polymerase, finds that end in the template, it just falls off. And the same here. There was a Taq polymerase coming this way, 5' to 3', and when it reaches that end, it falls off. So an original DNA template will give you a beyond pecan, but a beyond pecan will not give you something that goes beyond the sequence of interest. Except that you end up with, after only 2 cycles, what is still a bit of a hybrid molecule that I like to call semi-amplicon, because one of those strands is the true amplicon that you wanted, but the other strand is still a beyond pecan, right? So the beyond pecan, the hybrids between original strand and beyond pecans are infinite plus a beyond pecan. These guys are a beyond pecan plus half an amplicon. Okay? So after 2 cycles of PCR, we still don't have, strictly speaking, what we really wanted, which was that amplicon, right? There's still none. So let's do it again. Let's heat up again this thing.
So each time you're making shorter and shorter strands.
With a limit. The limit is now. Starting the 3rd cycle, you can't make him any shorter. Yeah? So for the 3rd cycle, we heat up everything separates, right? And then we go through, I'm putting them together now for visual simplicity, but we're doing annealing and extension. So after annealing and extension, now we have 3 molecular species. We have an original template strand with a beyond pecan, and on both sides it's the same thing. We have some of these hybrids, semi-amplicons with a beyond pecan and half an amplicon, and for the first time ever, we have amplicon molecules. They go exactly from one primer sequence to the other, from the forward to the reverse. Double-stranded DNA molecules, these molecules have 2 strands of DNA forming a double helix, the whole 9 yards. But they go from one position to another, and you decided what those positions would be when you designed those primers. Okay? Did that make sense? All right, we're not going to go through it in the same amount of detail, but if you did it for a 4th cycle, on top you have the template molecules, at the bottom you have the products. It's all color-coded so that you can later go and check what gives you what, right? But one thing that I hope you would notice is that now, after the 3rd cycle, we had 2 amplicons. I boxed them in a red line. Now we have 4. We have 1 here, 2 here, 1 here, except that it's not really 4, it's really 8. But we will get into that math in a second.
So an amplicon sequence makes another amplicon?
Exactly. An amplicon becomes the template for 2 amplicons. Because now, when we melt the amplicon at 95 and the primers find their target sequence, they will align precisely with the end, both ends of that amplicon. So now, instead of the primers finding their target sequence somewhere in the template DNA strand or somewhere, or rather one end of those beyond pecans, they will find their target sequence at the very end, one or the other, of the amplicon. So the amplicon serves as a template for 2 amplicons. Yeah? The reason we have 8 here is, as we will see in a couple of minutes, the math gets a little complicated, but we'll get there. The thing is that you keep doing this, and there are all these weird molecules that will accumulate every time that you do this hybrid between the template DNA strand and the beyond pecan, and these hybrids between beyond pecan and a real amplicon with those semi-amplicons. So they keep accumulating, but they will accumulate linearly with each cycle. Every time that you do this, you will generate an equal amount of these things, because it all starts from these really long molecules that you generated the first couple of rounds of PCR. After cycle 3, though, and as you were pointing out before, amplicons become the templates for new amplicons, and those become the templates for new amplicons, and you start getting this geometric accumulation, right? So, quick question for you. How many amplicons are produced by PCR for every original template molecule? 2, 4, we just said 8, so what's up?
Does it depend on how many cycles you use?
Exactly. Remember that PCR reaction is a cycle with many cycles. We actually decide how many cycles we do this thing for. If what we got from every DNA molecule was 2, 4, 8, we're not really getting many copies of DNA, are we? Like if we want many copies, what we do is we run these things several cycles. Starting at cycle 3, for every amplicon you have, you generate 2. Cycle 4, those 2 will give you 4. Cycle 5, those 4 will give you 8, and so forth. That's starting to look like a geometric progression, right? Base 2. So, roughly speaking, the number of amplicons for every original DNA molecule that you had is 2 to the power of, and here's a little number. We approximated to n minus 2, n being the number of cycles, and we subtract 2 because the first 2 cycles don't give us anything. It's only from the third cycle onwards that we get this geometric accumulation base 2. Okay, so if we run a PCR for only 4 cycles, that would be 2 to the power of 4 minus 2, so that's 2 squared, and 2 squared is 4, and that's what you see. 1, 2, 3, 4. Yeah? Now, I said that there is always some additional ones, and you see them here on the screen, so there's always some additional ones that I didn't box, but they're there. Now, those accumulate linearly, not exponentially. So every time that you run a PCR cycle, there's going to be these 4 new amplicons that come from those hybrid semi-amplicons that I mentioned before. So it's almost like they appear out of the blue, 4 at a time for every cycle. If you want to count their contribution to the total, you would do 4 times the number of cycles minus 3, because now we ignore the first 3 cycles. In the literature, you'll find all kinds of mathematical derivations of how you can calculate the number of molecules that you have at the end. You'll see all kinds of formulas. But I wanted to emphasize with you the following. Let's say that we take that approach. So if we run a PCR for 35 cycles, and you didn't have to know this, but I'm telling you, 35 cycles is pretty standard for a PCR, pretty standard. That number would be 2 to the power of 33 plus 128. 2 to the power of 33 is in the quadrillions. I don't even know. It's like insane. Adding 128, not adding 128 doesn't make a difference. So we normally ignore that term, and we simply approximate the number of amplicons to number of original molecules times 2 to the power of number of cycles minus 2. If these 2 to the power of 33 is a 1 followed by 14 digits, if I later add 128 or not, it won't make a difference. However, I emphasize approximately that number because, and this will come back to bite us, I guess, next week, because there is a physical-chemical limit to what PCR can do. On paper, we should get 2 to the 33 amplicons for every template DNA that contained our sequence of interest was present in that sample. In practice, that means that we would have to have at least 2 to the 33 molecules of primers. And 2 to the 33 times the nucleotides times the number of nucleotides in those amplicons. There is a physical limit to what we can add to a tube. So the idea that I want you to walk away with is this. Plenty of sources out there going over and over and over how many copies PCR gives you based on a given number of cycles. In practice, we never reach those limits. In practice, the reaction saturates before we reach those limits because we run out of primers, because we run out of nucleotides, because it gets so crammed in there that DNA polymerases can't even find the DNA, right? So in practice, there is a limit. And that has a consequence that we will explore next week, and it has to do with there is a point in which DNA, in which PCR is not linear anymore. Everything that we've described here, even though there is an exponential there, is linear to the original amount of DNA. The more original amount of DNA you have, the more amplicons you should have, right? Pretty linear. But at some point, that linearity breaks down, and that has a consequence for what we'll discuss next week called real-time PCR or quantitative PCR.